folks. I'm so sorry about the delay today. I hope it was conveyed to you that unfortunately I was behind an accident on the road. Um, I'm really sorry and thank you all for being so understanding with today. Um, just to check, has everyone sort of covered the topic of proteins and enzymes previously in their sort of classes, or is this brand new to everyone? Okay, so something that's probably quite familiar with people. Fine. So what I was thinking we could do is obviously cover sort of proteins and enzymes, but also think a little bit more about exam technique today, if that's okay with everyone. So are there any areas that people feel are particularly difficult? Thank you, Marcus. Let me know that in the chat that it's brand new for you. Okay, fine. So a few brand news then, which is totally fine. It's not a problem at all. Um, I will still try and integrate a component of sort of exam technique, but we will absolutely start with the more basic end of things. So I'm just going to start sharing my screen. Let me know when you can see that. Okay. Are we able to see the screen? Not getting any thumbs up, so this is not a positive start. Let me just pause the recording. Recording them. Great. So we're thinking about proteins and we're thinking about enzymes. Thank you all for bearing with. So we spoke about monomers and polymers, biological polymers, a little bit last time. We haven't quite spoken about lipids yet, but that's okay because we'll get there. Can someone remind me what the definition of a monomer is? Or a biological monomer? What is a biological monomer? Let's check chat. A small subunit that joins together to form polymers with the release of a water molecule during condensation reaction. Excellent. That's fantastic. Um, yes, so when we're talking about biological monomers, it's to do with repeating units. That's the sort of important part. It's repeating subunits that bond together, releasing a molecule of water to form a polymer, which is that sort of giant chain of repeating units. And the way that we then break up that polymer back into monomers is by adding water. Okay, so a hydrolysis reaction. Okay, perfect. Fine. So some important aspects of how we detect things. So remember last time I spoke about, or we spoke about even in terms of exam technique and testing for biological molecules. Does anyone remember what the key things to include in an answer are? We did talk about it. What did we say was important? Who was here last time? Let's have a look. Hey Laura, were you here last time?
I don't think I was here last time. It was my birthday last week, so I wasn't here.
Oh, happy birthday.
Thank you.
Fine, no worries. Let's see. Binta, were you here?
Yeah, but I kind of forgot um what to include.
That's totally fine. Can you have a guess at anything that we might need to include?
Um, where it occurs for the reactions, right?
Hmm.
Yeah, so where the reaction occurs and what molecules are being reacted.
Exactly. So really well done, Binta, and you've actually remembered more than you thought. So important things to mention when asked, what would you expect, or how do you test for X substance, let's say a carbohydrate? Well, you need to know what things you're putting together. So what reagent are you using to identify that thing? What conditions that needs to be under. So am I boiling it, for example? And also sort of what I expect to happen at the end. So if I get a positive result, what will that look like compared to the start? If it's a negative result, what will it look like compared to the start? And if I'm asked specifically about the bonding, I'm going to want to include specifically where in that molecule is that bond occurring. So we should all vaguely remember, I hope, when we discussed about the formation of the glycosidic bonds in your carbohydrates and the fact you can have a 1,6 and a 1,4. Now, to explain what that means, anyone who was here when we discussed that. Go ahead, Aki.
1,4 and 1,6 are the carbon atoms that the carbons bond to in the glucose structure.
Good. And how do we number them? Do you remember?
They're numbered from 1 to 6.
Good. Yeah. And does another numbering begins? Thank you, Akib. Fine. So that's one of your little pieces of homework trees is how do I number my carbons in that carbon ring? Because they'll expect you to draw out the monomers connecting to form a polymer. Fine. So as always, there are spec points that you can go over in your own time. That's not the specific test we're going to think about today for the proteins is our Burette test. Saying incorrectly, so I do apologise. But the spelling is now spelling is actually something that becomes more important in A-level biology because things are spelled quite similarly. So with your tests, make sure you do learn the spellings. Okay, fine. So proteins then. Proteins are otherwise known as polypeptides. They contain peptide bonds. It's why it's called a polypeptide. The monomer of the protein or the polypeptide is called an amino acid. Now, in humans, there are 20 amino acids and they all have a similar structure. Okay? They have to have an amine group and an acidic group, which is a carboxylic acid group or carboxyl group, to make it an amino acid. Okay? General structure is your amino group, which is your sort of completely gone blank. Your amine, if you think back to chemistry and amides, but also ammonia, almost like an ammonia ion. So we've got a positive charge on our hydrogen, and we have a carboxyl group with a negative charge on our oxygen here. Does anyone want to tell me something about, I've given it away ever slightly. What do you know about that? So, so first question is there a charge on this molecule? Go ahead, Laura.
Do you mean like about the carboxyl group just there?
Yeah, let's start with the carboxyl group.
So it's got, you're talking about charges, it's got a negative charge on the O. So it means that like potentially like a H could come over and like it could attract another H plus, H
A
plus.
H plus, good. What is
Sorry,
also known?
I forgot in biology it's called a H plus or a proton. I'm thinking about chemistry. But yeah, so then I guess it goes the same for the amino acid where there's the H plus on the end as well. It could attract an O minus. So that kind of means that it can form hydrogen bonds.
Good. And why do we care about hydrogen bonds?
I guess
Does
like,
anyone know?
oh, sorry.
No, go ahead, Laura.
I guess it means that like hydrogen bonds, like they're really, really strong. So first of all, yeah, they're strong, but then also it means that they can interact with water molecules as well. I guess that's more like for biology.
Excellent. So that is really important. And this comes back to your chemistry. So I'm actually quite glad that you started going off on chemistry a little bit. So when we think about biology, we're thinking on a big scale how things are interacting, but we're also thinking on a smaller scale. Now, the smaller scale is all about chemical reactions, so you have a certain level of understanding of chemistry here. Polar molecules will react with polar molecules. So if it's got a charge, it will react with things with a charge. If something doesn't have a charge, it's not going to react with it with something that is charged. Okay, so that's the first thing. Second thing is to do with sort of the reactivity of the group and the electrons available. So what is the availability or reactivity of this particular molecule atom? Okay. So as Alora pointed out, this O minus or this negative charge in oxygen leaves us open for the protons to be added on. Okay. And that becomes important when we think about our peptide bond formation. Okay, great. And about our hydrogen bonding, this then tells us how strong our overall structure will be. And we'll think about hydrogen bonding a little bit more shortly. But if there are more bonds between the molecules, the stronger something's going to be, right? So general structure, you have to have a carboxyl group, you have to have an amino group, you're going to have a carbon in the middle and a hydrogen, and then you've got this R group. Now, R groups will come up a lot. That just means replace with whatever. That's how I remember it, okay? So there are many different side chains or versions that can be substituted in here. And that gives us those 20 different amino acids that are available. Do have a look at your spec because different ones will require different levels of detail, but it's practice to, when you're discussing amino acids and bonding, to talk about an N terminus and a C terminus to the amino acid. It's saying which end has nitrogen and which end has the extra carbon on. So when we're talking about our bonding, it's between the N and the C terminus ends of the amino acid. Okay? It's being specific with your language. And we've said about the variable group here. Fine. Peptide formation then. Condensation reaction as before. And we sort of highlighted some of the points where this might occur. So it's between two amino acids, and if we have more than two, we're going up with a polypeptide. So dipeptide is two, poly is more than, or many. Now, this is representing one of the amino acids, probably glycerin, if I remember correctly. Now, if we're going to have two amino acids that are both glycerin, and we bond them together, we're going to form one molecule of water. Okay, so for every bond, it's one molecule of water. This is again just sort of representing that. Do apologise that we don't seem to have the lines drawn in. I will just do that very briefly. Fine. And those have obviously moved. Okay, so as you can see, at the C terminus of one amino acid, okay, one of the oxygens and the attached hydrogen group is going to bond with one of the hydrogens from the N terminus of the other one. So this might be confusing because when we first spoke about amino acids, we got this. What is special about this formation or form of the amino acid? Remember, there's a particular name for it. It's called a zwitterion. Perhaps you've not come across this, fine. I'll just type on screen. And it's named that way because it's the way that amino acids can be found in solution, okay? So it can exist as a zwitterion or it can exist as a non-polar substrate. Okay, so when it's our non-polar substrate, the difference is that we end up with an OH group. So it's an actual carboxyl group here. Okay, now amino acids are special in the sense that they flip between the state of zwitterion and neutral state. Okay, but the reason that we discuss it is to get you thinking about how it can react and therefore form those bonds that then make the important biological structures that we are thinking about. So I'll just give you that context so that it's clearer why we have this here. Okay. So we're losing a water molecule. We're going to lose an OH from the C terminus end of one amino acid, and we're going to lose an H from the N terminus end of another. So that's going to give us our water. We end up with a dipeptide here. Is everyone following this? Is it making sense? Yeah, great. I'm getting lots of thumbs up. If any concerns, please do let me know. Okay, fine. So we talked about specificity of language, we talked about terminals of the amino acid, and we talked about the variability that we have as well. We've also talked about the solution ions. Peptide bonds are formed by condensation. Conversely, they're broken with hydrolysis, as we expect with the rest of them. Fine. So we said that R group can be different, and glycine is one of the amino acids. It is the smallest one we have, and it has only an additional hydrogen. Okay. What else is special about glycine is it's the only amino acid that isn't chiral. Okay, so this is all the amino acids out there. You do not need to learn the different formula for all of them. You need to learn the generalized formula, but you do not need to know all of these. What does chiral mean? Good question. Any of our A-level chemists want to explain what chiral is? Go ahead, Elora.
Doesn't it mean that like there's 4 different groups around the sea?
Yeah, exactly. Thank you. So for carbon to be considered chiral, your central carbon, it has to be attached to 4 different groups. Okay. So if you've got your carbon double bonded to something, it's definitely not chiral because it needs to be attached to 4 different groups, and carbon only has 4 bonding points available. Okay. Now, if we look at glycine again, we can see it's got 2 of the same because it's got a hydrogen and a hydrogen. So that's not chiral. But if we then compare it to alanine, we see that there is a methyl group or CH3, a hydrogen, a carboxyl group, and an amino group. Does that make sense? Just check the chat. Akub, does that answer your question? Perfect. Fine. So we've got some important definitions, and I would bet my hat, although I'm not wearing a hat, that you'll get at least one question asking you to define one of these in your exams. So, got a helpful picture here. Who would like to have a go at verbalizing what a primary structure is of a protein? Go ahead, Anna.
The primary structure is just the order of amino acids in a polypeptide chain.
Excellent. And how is that dictated?
It's dictated by the DNA because the DNA like essentially codes for the mRNA, which can go to a ribosome to be made into a protein or polypeptide chain to be more precise.
Good. So the order of amino acids is determined by the DNA, and it's the sequence of what within the DNA that tells us the sequence of amino acids.
Bases.
Good. Yeah, exactly. So it's a sequence of bases and specifically the codons within the DNA that then tells us the amino acid that are being picked. Perfect. Who would like to have a go at explaining secondary structure? Go ahead, Laura.
It's the further folding of the polypeptide chain, which can form alpha helix or a beta pleated sheet.
Excellent. Okay, and how do they form those? What bonding do I need to know about for this level?
Hydrogen bonds. Sorry, I was just remembering it.
No, that's perfectly fine. You're absolutely right. So, important if they ask about secondary structure, you must mention hydrogen bonding. And the hydrogen bonding is forming between the R groups of the amino acids. So, we'll discuss what hydrogen bonding is shortly, but the R groups, those variable groups, will interact with one another because they're just right next to each other as proximity, and they'll form hydrogen bonds. Now, those hydrogen bonds will cause that chain of amino acids to kink or fold in a certain way. And we describe that structure at the end as either an alpha helix, so coiled, or beta pleated sheets. So that's from folding that's initiated by that hydrogen bonding. Okay, great. Tertiary structure then. We're getting harder, so I'd jump in now, and because if we don't get volunteers, I will pick. Okay, Nayan.
So there's lots of different types of bonds here. So like hydrogen bonding as well, ionic bonding. Um, I think it's called sulfide bonding. Um, and also interactions within the R groups that cause it to fold further.
Good. Any particular interactions you're thinking of?
I can't really remember that much.
That's totally fine. Thank you very much. So absolutely, your tertiary structure, again, you must include the bonding type that comes at this level. The bonds include your ionic bonding, your disulfide bridges, your hydrogen bonds, and hydrophobic forces. Okay. And again, this is all about the R groups. Okay. Those are the things that are going to dictate what the overall shape is going to end up as. That's the thing that changes between our amino acids. That means we can form those different types of protein shapes. And I'll ask a bit about how that relates to enzymes at the end. Okay, quaternary structure then. What's important about our quaternary structure? Alistair.
Um, I don't really know much about quaternary, but I know it's the folding of like multiple polypeptide chains,
Good.
but is it into like a larger protein complex and like tertiary? But
Good.
yeah, I don't know much else.
That's totally fine. So, as Alice said, your quaternary is where we have multiple peptide chains, polypeptide chains, that are folding together and associating to make a larger molecule, larger protein. Not all proteins do this. Some will stick at your tertiary level, but important ones like hemoglobin do have a quaternary structure. Okay? It's the thing that gives the final 3D conformation, final 3D shape. If we think about our initial amino acid chain, that's going to be 2D, it's flat. But we want something that has shape to it so we can then react with other things and break things down, which we'll come on to. Perfect. So this is just, again, words to what we've discussed. Primary structure is a sequence of amino acids that make up a polypeptide chain. It is dictated by the sequence of bases and codons within the DNA. It will be flat and 2D. The bonding between your amino acids is a peptide bond. Your secondary structure involves hydrogen bonding between the R groups of the amino acids within the primary structure. As we said, you're going to form alpha helices and beta pleated sheets from coiling or folding, depending on where those bonds form. So if you can't exactly remember what hydrogen bonds are, you can do a quick Google. I will also happily just explain it in a moment, so don't worry too much. Or even, shall we see if someone minds explaining it in the chat? So anyone in the group know what hydrogen bonding is and feel comfortable enough to explain? Go ahead, Akib.
Is hydrogen bonding essentially where you have where the hydrogen atom is bonded to a more electronegative atom, if you get what I mean.
Why don't you explain that for anyone who doesn't know what electronegativity is?
It's the power of an atom to attract electrons in a covalent bond. So essentially, it forms a bond with however many electrons there are. Essentially.
Good. Thank you very much, Akib.
No.
So, electronegativity, as summary, is the ability of an atom to pull those electrons towards itself. Okay? Now, because of the number of electron shells, number of protons, neutrons, all those internuclear forces, those dictate how much pulling force those protons can exert on any electrons. So remember, your proton's positively charged, your electron's negatively charged. This is not going to turn into a chemistry lesson, don't worry. But that means it's different depending on the atom because they have different number of shields, number of etc. The more electronegative atom is going to be partially negative in charge because it's going to have drawn those electrons away from sort of midline and more towards itself than the other atom. That then means that the other atom will be slightly positively charged. And the way that we denote this is with something called delta. So delta means partial. Okay. So here I've drawn water. Water has oxygen. Oxygen is more electronegative than hydrogen. Okay. And same over here. Okay. What you may or may not know, again, depends on not doing A-level chemistry, is that oxygen has two lone pairs of electrons. It will be using one of those electrons in its bond with hydrogen on either side, but it has some left over. Now, these lone electrons, or these extra bonding points that it would normally have, sorry, those lines are very, very small, also have a lot of negative charge because those are electrons. Okay? Something that has lots of negative charge is primed for attacking by something that's positively charged. Remember, opposites attract. Everything comes back to opposites attract. So the hydrogen can then form a hydrogen bond with the oxygen. Okay? So this partial charge is set up because oxygen's more electronegative. It can pull those electrons more towards itself in that bond. Okay? Oxygen has 2 lone pairs of electrons, i.e. it's got more electrons than are bonded to our hydrogens in water. So our hydrogen can then bond or form a hydrogen bond, which is sort of an intermolecular force, with that negative charge. So the positive charge is attracted to the negative. Does that make sense to everyone? If you're happy, give me a thumbs up. Great, lots of thumbs up. Fine. So with your hydrogen bonding, we'll look at some examples later, but you want to be always drawing it as a dotted line towards what other atom it is. Okay, that's how you denote your hydrogen bonding. It's a dotted line. Fine. So tertiary structure then. 4 different types of bonds. We can think about them here. So your ionic bonds, these are going to end up with electrostatic interactions between your ionic spaces, and they will contribute to folding. Okay. Remember, we're forming bonds in different places, and it's like pulling on bits of string that are in a big sheet. Okay, and you're pulling that bits of string, and you're ending up with the sheet folding in lots of different ways depending on which string you pull. And your ionic bonds are going to form electrostatic interactions. The disulfide bridges are where you have 2 sulfurs next to each other. So this is the rest of the amino acid. I'm not going to bother drawing, but if you have 2 sulfurs next to each other, they can form a bond and a covalent bond. Okay. The amino acids that would bond together for this are your cysteines. The reason this is important, does anyone know? Why do we care about disulfide bonds specifically? So it gives thermostability to the atom, to the molecule even. So if you've got a protein that has disulfide bridges in it, it will be more thermally stable. Okay. And hydrophobic, this is where we have non-polar amino acids. Remember, hydro meaning water, phobic meaning fear of, so it's going to repel. The reason for this comes back to our polarization of the molecules. So remember that water is polar. We just showed that image of it having a partial charge in different places. Okay, so only the parts that are polar within the molecule will be able to interact with it. Therefore, our non-polar bits will be repelled from the water because there's no way for it to interact. Fine. And this might be the final structure if the protein has a single polypeptide chain. As said, quaternary is where we have multiple polypeptide chains coming together, integrating to form a final protein structure. Now, the bonds that join this, you do not need to be able to name because they are various depending on what the overall protein is going to be, and there will be other associated molecules like cholesterol and so forth that might be inputted into the molecule to give it its function too. The important thing is this is the final 3D conformation. Okay. For anyone who's interested, hemoglobin has 4 polypeptide chains associated with each other. And I'm going to leave it there or I'll go on and on about that. Okay. This is just to say about the number of chains that might occur. So if you've got 2 polypeptide chains, you'll have what we call a dimer. So think polymer, but we're calling it dimer because it's 2. We can name something with 3 chains a trimer, tetramer for 4, and so on and so forth. But typically you'll only really be expected to mention the word dimer, if anything, or polymer. We said it previously with our carbohydrates, but you can have homonomers or heteromers. Sorry, I can't even say it. Heteromers, and the difference is whether or not they have different peptide chains associated with different ones, or if they're the same. So going back to my hemoglobin example, there are two so-called alpha chains, which are one type of chain, and two so-called beta chains, which are a different type of chain. So it would be a heteromer words. Okay, fine. And again, this is just building up that language that we've already explored. Fine. Any questions about that? Things to know for exams is the definitions for each level of protein structure, so primary, secondary, tertiary, quaternary, including the bond types that are there or involved in each stage. Okay. And then also thinking about how that might link to mutation. That's really important, and they will often make those synoptic links in the questions too. You do need to know the general structure of amino acid. I wouldn't be surprised if they asked you to draw that. Okay. And to be able to appreciate or indicate where those peptide bonds will form. Because again, they may ask you to draw a box around where those bonds form, or they may ask you to describe it. Burette test then. This is for your proteins. We helpfully have two test tube images on our right-hand side. We have the pink one, which is saying there's protein, and a blue one saying no protein. So these are the results we're looking for. And again, remember, when you're writing your answers, you want to say what the test is for, what conditions you're using, what reagents, so things that are reacting, and then what positive and a negative result would look like. So to start, we need to make the solution with our sample alkaline. So we add a few drops of hydroxide, copper sulfate to apologies for that, folks. Don't know what happened there. So you must include the positive and negative results if the reaction did or did not occur. Okay, great. Fine. So let's think about function then. I've been going on and on and on about how bonds relate to shape. But how does shape relate to function? Anyone want to have a guess? This goes back to your GCSE biology, and you can use any of the examples on the screen to help you. Go ahead, Naomi.
Like for enzymes, the shape of the active site for substrate to come in and be broken down or built up or something.
Good. Tell me more.
What more do you want to know?
So what I would like to know is, so you're saying that that's going to determine the shape of the active site. Why does that matter, first off? And what happens if I change that shape?
So if you change the shape, then the substrate will no longer fit in because it's very specific to the shape of the substrate, the active site.
Excellent. So if I have a mutation, what might happen?
The shape of the active site might change, but most of the time it's not on the active site.
Good.
The
And
the
if I
where
had,
the mutation is.
sorry, no, go ahead.
Where the mutation is, like half the time isn't on the active site.
The answer is yes. So again, think about your sheet, okay? Sheet with lots of threads in it that we're pulling, and it's going to change the shape. Now, if I pull the threads on one end, that end is absolutely going to change shape, but it will also have an impact on the other side of that piece of fabric because I've still pulled on those bonds, essentially. So if I have a mutation that changes another part of the enzyme, it could still, I'm not saying it will, it could have an impact on the overall shape of the enzyme, including the active site. So if they give you a question where they say, we have a mutation, it's not in the active site, but in this reaction, the enzyme no longer works or the reaction is no longer happening, explain why. It's to do with the change in the conformation of the tertiary structure, the bonds, where those bonds are taking place, changing the shape of the active site still, and therefore making it not be able to react with specified substrate. Does that make sense? Thumbs up if we're okay with that. Great. Fine. So that's enzymes. What about antibodies? Let's go with Bernadette. Are you able to unmute? Or Sophia? Check. Oh, that's fine. Don't worry, Sophia. Thank you for letting me know. Okay, fine. We'll move on. So, as we said, enzymes have a specific shape. Think about that lock and key mechanism you were taught in GCSE biology. Your substrate needs to fit into the enzyme active site. It's very specific. Therefore, if there's a change in active site, there's a change in function. Similarly with antibodies, they have an active region. So we'll quickly go through this. This is an example of an enzyme. This is your enzyme substrate complex. Another key phrase, so if you're asked about enzymes, make sure you're using that phrase, enzyme substrate complex. Your antibodies have a constant region and a variable region, okay? And so that's going to have specific antigens that it can bond to, dictated by the shape of those regions. If we change the primary structure of that protein by changing the base sequence in the DNA, that's going to impact the secondary, tertiary, and quaternary structure of the protein and change its function, potentially, unless it's a silent mutation. Okay. So as if we have a constant region and we have a variable region, and that's the part that hooks onto our antigens. That gives specificity to the protein. Any questions on that? Do people know about ELISA, the ELISA test? Okay, we'll come on to that later then. No, fine. We'll think about LIFO shortly then. Fine. Transport. Proteins have specific shapes. We dictate that specific shape by the base sequence. I'm repeating myself because it's all the same. Within your cell membrane, you are going to have proteins that allow the transport of certain molecules, including water, because water is polar. And remember, our phospholipid bilayer is hydrophobic. Okay. So we have our protein channels. We even have protein pumps. Okay. And they're all going to have specific shapes to be triggered by certain things or to transport certain things. These may be those that require the substrate to bind to the protein in order to change the shape of the transport protein and then pass it through. Okay? And that's sort of a hinge mechanism. What we see here is an example of an aquaporin. Aqua meaning water, porin meaning pore, as a little hole. So this allows water to move across the cell membrane. And it's an example of our transport proteins. Fine. You also have structural proteins, so things that give support or physical form to cells and organisms as a whole. So we can think about our mitotic spindles in mitosis. Okay, those are formed by proteins. We can think about on a larger scale keratin in hair, collagen in cartilage, that's between your bones. That acts as sort of a cushion so that your bones don't rub against each other and cause damage. What else is there? There's pigment, protein pigment in sort of skin and in eyes as well. All these different things that proteins do. It's pretty amazing. And they will have covalent bonds within them. You think about your peptide bond, that is in itself a covalent bond. And then other levels, so your secondary, tertiary, quaternary will also have the possibility of a covalent bonds forming. And this is going to allow those long polypeptide chains to link strongly to one another and form those specific shapes. So you have more covalent bonds within your structural proteins and perhaps within other proteins because you need that strength to maintain the structure more so than potentially others. So these are your sort of key points. Again, I'm not going to go over them. I'm not going to label the point, but remember your tests, you need to mention what it's for, conditions, reagents, and the expected result for a positive and negative. Okay. Fine. Do I want to go through this? Not in huge amounts of detail. I think people are quite happy that enzymes are biological catalysts and they act by lowering the activation energy. What they do is exert sort of mechanical pressure and chemical pressure on the bonds within the substrate to then allow it to break or whatever it might be. Fine. We've said about this already. So, potentially basic question. What is an active site? So I'm going to give you 1 minute to answer this. We're going to take the 1 mark question. What is an active site? You want to put your answer in the chat once you've done it? I can then mark it. Excellent. So really good answer coming in that are using key terminology. So think about what key terminology they might want you to use in your exams. Good. Fine. So for your mark scheme, you're going to want to include that it is the site of the enzyme or the area to which a complementary substrate will bind to form an enzyme substrate complex, and that these are complementary in shape. Okay, so an area of an enzyme that is complementary in shape to a specific substrate that allows the catalysis of a reaction. Okay. Good. I won't ask you to do that because we're going to move on to more of an examinary type tutorial. Have a go at this one. And again, it's probably very quick. And I believe we even possibly did this previously, so I'm expecting full marks. We got any answers? Just check the chat. Good. For bonus points, try and use the terminology that I mentioned earlier about your terminals. Okay. See if you can integrate that. Good. Excellent. Fine. So we have gone through it before, so I'm not going to spend too much time. So is your condensation reaction between the amine of one amino acid and a carboxyl group of the other, or between the N-terminus of one amino acid and the C-terminus of the other? Okay. Excellent. Fine. So again, for your bond formation, you must include the location. Okay. So you want to think about what reaction type, where. Next question. Okay, have a go at this. Okay, so once you have an answer, do please put it in the chat. Let's have a look. Good. Great. So remember, specify where. If you're going to mention a bond, you want to mention where it's occurring. Okay. Fantastic. Good. So we'll show you a couple of answer types. So we've got one option. I want you to tell me how many marks you might give this, okay? Okay, so I'll read it out. The secondary structure contains multiple kinds of bond. For example, hydrogen disulfide ionic bonds. What do we think of this answer? Got a couple of zeros. Yeah, so this is tricky. Unfortunately, this would probably end up as a 0 because they've confused secondary and tertiary structure. Okay, so they've included too many bonds in their answer, and they've not actually mentioned some of the key structural properties of secondary structure. So here's another one. Secondary structure is simple folding of the protein to form beta-pleated sheets. This structure is held in place using hydrogen bonds. How many marks would you give that? Okay, we have some contesting in here. Yeah, good. Perfect. So, if I'm going to be really picky as an examiner, they've only mentioned beta-pleated sheets, they haven't mentioned our alpha helices, and they haven't said where those hydrogen bonds are, which you've all told me is between the R groups. Now, I would not risk it. I would include that detail, and I would only give this 1 mark. The examiner here, and this was a little while ago, so it may have changed, gave them 2 marks. I just would not risk it. I would get in the habit of saying this structure is using these bonds in these locations to form this thing. Okay? And if you can get that into a succinct sentence or a succinct set of points to then repeat every time you get this sort of question, you'll be way safer in your exams. Okay? And again, some students unfortunately fall into the trap of confusing secondary, tertiary, and quaternary structure of proteins, but also confusing the difference between protein structure and nucleic acid structure. Read the question so carefully. Okay, write your answer and then reread the question. Even if you lose time because then you realize, oh, I've answered the wrong question, at least you've picked it up and may still gain some points. Okay. Fine, I believe this might be our last question. Let me just double check. Yes, our last question for today. Have a go and put your answer in the chat. So be careful here. Okay, it says that we have the same type of amino acids. That means the same R groups are present. And a few of you have, I think, misunderstood that. So just have a quick think about that. Okay, because it's late and I do not want to make you wait any longer, I am so sorry again for the delay today. This is a 2 marker, so it should only take 2 minutes theoretically. We're being asked to explain, therefore we're saying something, we're stating it, and then giving a reason. Okay? So state facts, give reasons. Same type and number, but different tertiary structures. So it'd be good to explain what tertiary structure is, potentially, and think about what is happening in a tertiary structure and how that then relates to the amino acids. Okay? So first example response. Proteins are able to have the same number and type because they have bonds in different places and look different. What do we think about this answer? How many marks would you give it? So I've currently got one response saying 0. Any other takers? Okay, it's 2 for 0. Any others? Okay, here's an easier way. Thumbs up if you think 0. Thumbs up if you think 1. Thumbs up if you think 2. Great, so I actually agree with the 0. And again, this might be really harsh, but you want to be more harsh when you're doing your practices anyway, so you can maximize the likelihood of you getting points in the exam. So the reason is, although they have the right idea, okay, because we can have a different order of amino acids and therefore different order of our bonds and so different formation of different things, they haven't used any real scientific language here, okay? They haven't actually said amino acid at any point. They haven't related it to the bonding type, so what bonds are we talking about? And so I can't give them any credit for it. Have a look at this one instead. So proteins are able to have different structures because of the sequence of amino acids. Change in primary structure means that the ionic, hydrogen, and disulfide bridges form in different places. This results in different folding and a different overall shape. How many marks are we giving this? So I take your point that they've potentially not mentioned where those bonds form. So do mention the R groups in your own response, okay? But actually for a 2 marker, could I give this to them? I think I would, because I think it would be a 3 marker if I wanted to have a bit more detail. So the reasoning is because they've mentioned the specific information about the primary structure or sequence of amino acids and the type of bonding present. I would want to include in that second point, again, if I was being a harsh examiner, to say ionic hydrogen disulfide bonds between R groups in different places because of that different order. Does that make sense? Yeah, great. So think about the language you use. Okay. As you can see with your explains, you've had proteins are able to have different structures because of the sequence. So we're stating it. We're then linking it back to the question. The change in that means we've got a change in bonding. So that's my reason, which equals what I've asked. Yeah? So statement, reason, question, linkage. That's your explains. Statement, reason, link to the question. I hope doing these sort of exam-based questions are helpful in your technique and you are finding them useful. If it's not, please do let me know. I'll stop the recording there.